Assalamu alaikum dear students. Today we will discuss about very basic topic, that is transcription factors. We will learn about its basic definition and its working. So the topic is transcription factors, and whenever you are starting a topic, try to know the meaning of the words or the name of the title. So here it has two words, transcription and factors. So transcription, we know that it is a process in which the information coded in a sequence of DNA is converted into the sequence of RNA, and it is very important, or you can say the starting point of the central dogma, which is the process required for the gene expression. The next word is factors, and a factor is any element that has an influence of or effect on any process or condition or situation, that they play their role in one way or the other. So transcription factors are such elements that influence, that affect the process of transcription. So transcription factors are such elements which play their role in the process of transcription. Now if we talk about the chemical nature of the transcription factor, we know that all biological molecules have been categorized into carbohydrate, proteins and lipids. Among those, transcription factors belong to the group of protein, that is, they are protein in nature, they are made up of amino acids. So now you are able to define transcription factors. Transcription factors are the proteins that regulate the process of transcription. Now pay attention to the word used here is regulate. It doesn't mean that they work in one direction. They can either upregulate the process of gene expression, or you can say the process of transcription, which is one of the steps among others in the process of gene expression, or they can down-regulate the process of transcription. Now question arises that how transcription factors upregulate the process of transcription and act as enhancers. We know that in non-dividing cell, DNA material is present in a form of chromatin, which is aggregated as shown in this figure. In this conformation, general transcription proteins cannot recruit or cannot bind to the, you can say, the promoter region or the transcription start region where they have to bind to start the process of transcription. So what do enhancers do? That they come and bind to the DNA, to the DNA sequence particular, or they can be non-specific also. So they bind to the DNA and change their configuration by modifying the histones and thus influencing their location in the gene. Now as you can see, now this thread-like structure seems to be unconvoluted or less aggregated. In this conformation, these general transcription proteins are now able to come and bind on a promoter region and can start the process of transcription. So, and we know that initiation of transcription is a major step in expression of any gene. So here in this way, this transcription factor has act as enhancer and enhances or initiated the process of transcription. So this next question is that how do transcription factors downregulate the process of transcription? What they do that is that normally what should happen, that transcription general proteins should come and bind to the promoter region and should start the process of transcription. But what happens is that the repressor might get into the closer to the gene, the promoter region, and binds there, because of which they physically block other important transcription general proteins to come and bind. So in this way, by physically blocking the general transcription proteins, transcription factors act as a repressors. Before going to this step, I would like to make it clear that have you noticed that enhancers has enhanced the process of transcription by having a chemical influence, while repressors do so by physically blocking the transcription proteins. So next is that how do transcription factor do that? Okay, what happens is that transcription factor proteins have this ability because they have a particular domain, that is DNA binding domain. This domain can recognize and bind to the single-stranded or double-stranded DNA. So these domains, there are many such domains which have been discovered till now. Few example is helix turn helix or zinc finger or leucine zipper. These transcription factors, the elements or the sequence at which these transcription factors bind are called cis-elements. So here I have a question for you, that are transcription factors only protein category that have DNA binding domains? I would like to see the answer from your side in the comment section below. Let's move on. So application of engineering transcription factors. By modifying transcription factors, or you can say by engineering them, we can influence a large number of reaction at once. That is, a single transcription factor is not associated with a single reaction or a single process only. They are associated with many others. So in this way, by modifying one, we can affect many. Another application which I would like to discuss here is that how we can use them in uh, improving our crops. What happens is that if we get to know that which transcription factors are actively expressed during the stress condition, we can take them from those plants and can add them into those which are agriculturally important for us or which are economically important for us. So in this way we can manipulate, you can say humans are always manipulating nature, okay, although humans cannot. In anyway, so transcription factors in this way play their role in improving the cultivated plants, which are either the food or important in textile or in one way or the other. Another important application of transcription factor is that nowadays it is being used for screening transformed plants. We know that after doing genetic engineering on any organism, we need to find that which organisms or individuals or strains or species have been transformed and which are still not. So in case of plants, it is a bit more laborious process because it takes more time than doing it on bacteria and other. So here is another solution provided by transcription factor. What do scientists do? That they check the expression profile of a transcription factor in non-transformed plants, and then they do it for the transformed plants. So by comparing by screening this expression profile of all the progeny, they can tell that which plant has been transformed and which has not been transformed. And another way is that to get desired control on gene, yes, of course, transcription factors is a main thing, or you can say the first thing that controls gene expression. So we can manipulate transcription factors in this way, and scientists are also doing research or work on increasing or manipulating the biosensing capabilities of bacteria for us, because the transcription factors are triggered or they are expressed in return or in case of receiving any stimulus from the environment. So in this way you can say that by changing that, we can, or by checking that which transcription factors are being responding to which stimulus, we can manipulate it. So why transcription factors are so important? If you want to know its answer, I think you will get to know it if you go for why gene expression, or why different gene expression at different stages of development or at different environments or whatever. Why we do need different gene expressions at different times or at different stages or in different environments. You will find the answer why do we say that transcription factors are that important. The next thing is, what does it mean then? So we differ in transcription factors regulation only because it is only transcription factors that are making different gene expressions, and that is why we are differing in phenotype type. Oh, please don't forget alleles. We have alleles. We have different alternative form of genes too. So both play their role in showing different phenotypes. So the thing is, after that why nature has chosen proteins to act as transcription factors. Getting my point? Why don't nature thought that the carbohydrates should act as transcription factors? Why in the process of evolution, why in the process of whole of the generation of this life, protein, the category of the molecule, chemical nature of the molecule, protein has been chosen to act as transcription factors? So looking for good discussion in a comment section. We'll see you with more topics. I hope you have got some idea from this video. Allah Hafiz.